A warm welcome, good morning. Second day of Abu Dhabi Global Healthcare Week, and today we have a very special topic. We're talking about rationalism in the hype of large language models, and to support us or not, OpenAI yesterday announced a new GPT model, and of course that is a big hype in the media. And today Google has its house fair, IO, and most probably they will talk about large language models. And of course, that is the perfect framework for our humble panel discussion about rationalism in that hype. And I would like to give a warm welcome to Alex Aliper, the president of In Silico Medicine, and Martin Hofmann-Apitius, professor at the University of Bonn, and also in a very important role at the Fraunhofer Institute that maybe you guys here in Abu Dhabi do not yet know, but soon in the future. So, welcome. So there is a large hype, and recently Martin and his team has published a remarkable paper investigating the capabilities of two OpenAI GPT models. And there were some, let's say, surprising results, surprising insights, and maybe Martin, could you come up with some of these insights and your opening statement?
Yes, thanks, Rudi. First of all, since we are talking about large language models and knowledge graphs, and I try to have an overview on that topic. This is extremely moving fast, and everything we discuss here may be actually obsolete within weeks and a few months or so. I have never in my entire life as a scientist seen anything that is so dynamic so fast, honestly. And I'm doing that for a while. So that was important. What are we doing? My team is actually working on turning textual knowledge into computable knowledge, and that we do that by turning what we know as scientific papers into computable graph models. And these are knowledge graphs, and that's a big part of our work. And we do that actually at the level of complete indication. So we work on indication-wide knowledge representations in graphs that you can compute on. And what does that mean? You can run algorithms over it, and you can confront this knowledge with data. You know, you can make data and knowledge interoperable. Now, when the LLM hype started, everybody was impressed, and I was, of course, impressed too. And I think everybody really played with ChatGPT and so on. We have seen that the large language models actually can do this job of extracting of knowledge and representing it in the form of a triple, which is the knowledge atom, so to speak. And we tested that. And we were running it against a special trained model, a BERT model that was optimized for triple extraction, and did a comparison. And this is the paper that Rudi refers to. And I think that was just one of the studies that did the systematic comparison. We will see by far more in the future. But I stop at this point because if I start talking about these benchmarkings and so on, I will never stop. So I hand over back to you.
So, Alex, the good old question, but I think especially your company is pretty much into that, yeah, let's say hype, using AI for drug discovery, maybe drug repurposing things. What is your perspective on that large language model hype?
Sure. Well, it's great to be here, first of all. Thank you for the invite, Rudi. Our company has started more than 10 years ago, back in 2014, on Johns Hopkins campus in Baltimore. And that was the dawn of AI. Like, it was called deep learning back then, right? So, and it gradually evolved into being more kind of encompassing and comprehensive tooling. So back then, we immediately started working with generative models. We published our first paper in generative adversarial networks and autoencoders back in 2015, first peer review paper in chemistry. And we also started applying text to text. So when transformers came out, right, so we kind of started playing with them, and we kind of saw the limitations. And the BERT framework that was released shortly after that was adopted quickly, and we also started exploring, and we immediately saw the power and the importance of benchmarking. So we must always benchmark our models against each other and against humans. And for chemistry, we developed a framework called MOSES, molecular sets. So that framework allows you to benchmark generative models against each other by using like several metrics like diversity, novelty, etc. So we have the same framework for knowledge extraction from text, and we have been kind of witnessing quick progression of those models in terms of accuracy, in terms of even like novelty. Sometimes some components of extracted knowledge were surprising to humans because they missed their attention. Of course, they were like wrong. Some of the pieces were wrong, but some of them were surprising and interesting. So we adopted it quickly, and knowledge graphs, biomedical knowledge graphs, quickly became a part of our platform because we have 3 tools in the platform: the tool for biology called Pandaomics, the tool for chemistry called Chemistry 42, and for medicine called InClinico. And for Pandaomics, we have a separate biomedical knowledge graph, curated and reviewed by humans, but initial knowledge extracted from public domain using NLP methods, and humans are providing QA/QC. So we have witnessed the progression of those models, and you can like see even recent benchmarks, right, that outperform BERT by like large margin, or other like mapping technologies like REACH or IDOS and others. GPT-4 is by far the superior method. Of course, it hallucinates, it has drawbacks, as was indicated in the paper, right? But I think the entire industry and the entire field is quickly getting closer to human level accuracy.
I will come back later to that, and I will ask you about the sweet spot thing that I've mentioned previously. So Martin, could you walk us through some remarkable insights of that paper that we mentioned initially?
Sure. One of the things I should make clear is that we are looking into a very special form of knowledge. We look into cause and effect relationships, which is a subset of all the triples, all the knowledge atoms that you can extract from text. That makes things a bit more demanding on the side of the model, and we have a little framework that allows us to systematically test at the syntax level whether a machine actually does performs well in the extraction task. And we found a couple of things with the GPT models, that they are not as good with resolving objects. The reason is that in text, we have, for an average human gene, we have our protein, we have about 100 names in the literature. So there's synonyms all over the place, and the synonym resolution is something that comes partially automatically in the training process of large language models, but it's not perfect, and that shows. And that becomes highly relevant later on when you want to do mappings between objects in your knowledge graph and data. And that was one thing. The other aspect was that we had, with our special syntax that we use, these cause and effect relationships that we focus on, we had a very special training set for the BERT model. And that was really optimized for that. It had a bit of an unfair advantage, although I should say, funny enough, if you ask ChatGPT, do you know BEL, the biological expression language, the syntax that we use? It actually says, yeah, I've been trained on that. And they have been training ChatGPT on our documentation for my institute of BEL. So it takes everything from everywhere it knew about our syntax, which is not so widely used, I have to admit. By the Harvard people and the Indra people, for instance, they use Bell. So the big mechanism, big mechanism initiative in the U.S. They actually had used Bell as an output, and that's mechanistic knowledge representation. There's a Harvard group with Phil Sorger, who is they are actually producing this sort of content. So it's not that we are a total orchidee, but we are, with our syntax that we use, we are a bit special, which makes things for a generalized model a bit more demanding, I would say. Coming back to your original question, in our case, the special trained BERT model was still outperforming the GPTs. We would actually need to do it again, the experiment now, because as Alex said, and rightly said, this is developing and improving so fast that you can do these benchmarks actually continuously.
Yeah, well, I think I see already the next manuscript and paper I had. So you mentioned that unfair advantage and playing back that to Alex. You're into medicine, into biology, and into chemistry, and you started with chemistry, which I found very smart, as in my humble private opinion. I always thought, well, okay, chemistry is definitely more precise than the rest of the biology. But now asking, is there a sweet spot for knowledge graphs and LLMs in comparing medicine, biology, and chemistry from your perspective?
Well, this is called biomedical knowledge graph, right? So because you have different types of entities linked to biology, like genes, proteins, pathways, different relationship between those. You have chemical sphere like molecules, drugs, and you have medicine, actual clinical trials, clinical studies, preclinical models. All those relationships can be captured in a single graph. And we actually trained those graphs to predict certain properties. In our tool in Clinico, we're predicting the likelihood of transition from phase 2 to phase 3 stage human clinical trials. And this is typically like 2 out of 3 trials, they fail in phase 2. And this is a crescendo moment for all clinical development efforts to see if a drug is efficacious or not. That's where you test efficacy. So we build a model, transformer model, on this biomedical knowledge graph that captures relationship between molecules, biological pathways, and clinical studies. And we're able to get very reliable performance of like ROC-IUC of 0.8 and over, depending on therapeutic area, in the prospective, quasi-prospective validation study. So I think like sweet spot is going to be combining different realms, so to speak, of biology, chemistry together to elevate the level of reasoning of these models. And further on, maybe we'll touch on this on a kind of closing remarks. I think foundational models that capture the basic concepts of biology will be gaining more and more prominence in the future.
Okay, yes. Next one.
No, I just wanted to directly respond to Alex's point, because he mentioned the biomedical knowledge graph and pathways, and that's what we all know. Pathways, we know pathway databases and so on. What we did in our paper was actually looking into a very specific post-translational modification event in a certain part of a protein, which is the p-tau molecule, which is one of the hallmarks of neurodegeneration. And the pathophysiology of neurodegeneration is really around these subtle changes in patterns of post-translational modification, of phosphorylation. So we're going into a level which is way below what you know from pathways and from, you know, this gene is important, that gene is important. And I think this is also illustrating one of the next frontiers that we're facing, Rudi and Alex, because we need to go much deeper than what we had with the common knowledge about the regulation of genes or the interaction and the signaling that you observe in the fibroblast or in an immortalized fibroblast or in the cancer cell. And when you really dig deep into pathophysiology of diseases, then you end up in highly complex relationships, and you need an optimized system really to deal with that. And even the identifier that I was mentioning, like, it doesn't give you really the information you need if you know the identifier for tau. You need to have the position of the phosphorylation acceptor sites and so on. So it becomes much more finer granular. I'm very optimistic that we are getting there. So I fully subscribe what you said, Alex, on the ramp up now that we observe with the large language models. But I just wanted to raise awareness that we also leave the time of KEGG and reactome and wiki pathways and how they are called behind us in precision medicine.
Alex, let me come back to that medical area, and sometimes it seems to be a tiny elephant in a big room or a big elephant in a tiny room. That is clinical phenotyping and data quality and data curation. So I think, okay, large language models, we need training data. And what is about the precision of the training data and data curation? How is your perspective on that?
Great question, and very important topic, right? In clinical realm, you very rarely, so there is an intrinsic bias. Pharmaceutical companies, they typically do not publish results of the trials that fail, right? So there is a bias towards success. And only internally, those pharma companies can process this, like the full scope of information, and build reliable models to, make inferences on why it failed, do a postmortem analysis clinical study. So there is a bias issue. There is an issue of quality, data quality, right? So data sometimes is misreported. You don't have sufficient sample size. You make errors in interpretation, and that kind of snowballs, right? So having a workflow for QC/QA with human experts is essential when preparing your models for medicine. That's what we did with our tool. And in general, that's a workflow we adopt across the platform. So there's always an expert that oversees the end result of the workflow and puts in a stamp of approval if it kind of meets the basic criteria we have SOP on. So it's a really important topical question, and I would encourage industry players to publish more data openly, right? There is already an initiative by the FDA to encourage companies publish actual failed clinical trial data. It is a big problem, and the only solution here is more transparency, collaboration, and I would say integrity, basic integrity across the entire industry.
Martin, I see you're moving already.
No, I just want to say that we see also a impact of large language models now in authoring, and in even increasing the flood of papers. And there's also, when we talk about knowledge graphs and papers where we get information from, we need to be really careful in selecting the right literature. We do also, a triple index of the entire PubMed. So this is possible, but we always need to be aware that people publish a lot of rubbish. They exaggerate. You all know Johnny Ioannidis at Stanford, who is regularly bashing the science community for essentially misconduct and inflating results and exaggerating and generalizing where you can't generalize and so on. The big chance that I see actually with the knowledge graphs on one side, or the computable graphs as I called them in the beginning, and the collection of large numbers of data, is that we can build what Barend Mons, a colleague from the Netherlands, some while called the knowledge collider. It's like the hadron collider, like the particle collider. You have data, and you collide the data with knowledge graphs. You build essentially discovery engines that do the job for yourself, because you can't look into all these subgraphs and all the interesting patterns. You need machines that automatically run all the data against all the knowledge, essentially. And this is something I would really predict. I'm usually careful with predictions, but we will have AI-based discovery engines that do a good part of the discovery process autonomously.
I would just add that it would be actually amazing to have a AI-based, large language model-based academic policeman that would go after exaggregated claims automatically.
So, okay, this means we now have to invent the hashtag academic policeman or AI-based academic policeman. But that is really a good idea. We should post that later. So let's stay a little bit with that thing. And I think there is someone coined that thing of replication crisis in biomedical science. How much does that affect us? So from my previous practice, I know pharmaceutical companies trying to replicate academic publications, and they failed in around about 80, sometimes 90% of all approaches, which is a bloodbath, of course. So does that affect your work, Alex?
That's a reality we have to kind of navigate in, right? So we are not only aware, we have tools to detect conflicted claims and something that has higher chance of being, low quality, reproducible. And that happens not only in publications, we see it all over the place in patents, right? So many companies, they patent stuff that couldn't be reproduced. Sometimes they do it on purpose, just to throw people off, and you wouldn't be able to replicate the synthesis of a specific compound. And yeah, that's sometimes done on purpose. So I think there is a component where there is a real kind of complexity of biology that contributes to that conflict of this misunderstanding and conflicting data signals. But sometimes, yeah, it's conscious bias and conscious misdirection by the authors that must be acknowledged, must be flagged, and I think penalized ultimately. We have tools to deal with that, work around that, but it is a problem. And right now, academic publishing is a big business, like billions of dollars go into that, right? So it's huge business. So more and more papers coming out, and it's paramount to have an arbiter that would assess the quality, the claims, the reproducibility, where the data corroborates with previous knowledge, and again, transparency, open science is something that should be emphasized by the editorial boards of those journals, right? So we should have more reproducible papers out, especially computational papers.
Okay, bioinformatics can do a lot of harm, but sometimes the researcher might be innocent. Let's say we talk about the data class of gut microbiota, for example, where they rely on the lab to a proper DNA extraction. But the lab can produce tons of confounders, be it Lisa, the technician in the lab, or the bioinformatics you apply with background databases and their setting just when you go, let's say, for phages, and you play a little bit with the settings of the background database, and suddenly you discover species or something else and strange. Yeah. Where the researcher then, who is maybe not the lab pro, but relies on that result, suddenly sits on a data set that is, let's say, problematic.
Not only that, I think Broad Institute once, they discovered that depending on the time of the day, they take omics data measurements. They have different signal and response. There are fluctuations based on the almost like gravitational field, right? So it's complex. Biology is hard.
And it's not only the timing, it's also the temperature. So you have rodents from the same genetic makeup, 21 degrees, 24 degrees, you already have a different microbiome in the rodents. But
if somebody is walking in the lab, they create vibrations as well.
and we train our models on that data, where we literally have no idea what was the temperature, when was sample sourcing, and so that is some kind of noise. And suddenly we produce artifacts and train our models on that. So obviously that is an issue. What you screw up during sample or data sourcing is a problem. Martin, you're a big fan of something else, that is precision psychiatry, and let's say clinical phenotyping. And here the elephant might be the precision of the clinical phenotyping. And what impact does that have on knowledge graphs and large language models?
Well, that's a non-trivial question. What I can say is that the assessment by questionnaires, for instance, which is quite typical in psychiatry and also in neurology, that people try to find replacements for rather individualistic or subjective responses, which also, by the way, vary with the time of the day or the mood and so on. I've seen data in Lausanne from a memory clinic where people were actually reverting from an Alzheimer diagnosis into normal. And that can only be explained by a major depression or whatever, so in between. So the psychiatry field is a minefield. And
I mentioned it.
and what the current idea is that people say we do speech, voice pattern recognition, and we monitor also the usage of these devices here, because our interaction with machines actually has also steeply increased, and the machines can keep write actually nice records of that. So the current hope is that we can at least partially replace the older procedures, which are also, by the way, just occasionally. I mean, you have a visit and then you have a person with a snapshot, and you don't do continuous monitoring, and everything now is going towards continuous monitoring. There's one thing I wanted to say in response to Alex or on the topic that you just discussed, because I'm a university professor and I teach, and I don't know a single curriculum in the world where data quality assessment or self-critical reflection on experimental planning and on how you choose your appropriate analysis strategy, how you ask yourself whether this already generalizable or should I better be more modest. This is not trained. It's hardly trained. I mean, if you are know of a curriculum where that is done, I really appreciate the pointer, but I can say, unfortunately, the university I am with has no dedicated, philosophy of science. That starts really with a philosophy of science, and then proper data scrutiny, and SOP scrutiny and error models.
So you don't have a lab book for that. Okay, Alex. Now we see the role of the knowledge graph and large language models and the overall headline of AI hype. And that also comes with not only narratives, but also equity stories of companies. And there is a wide jungle with many people doing all kind of stuff, claiming of, let's say, we have that super model. Most of the time they say it's proprietary. If that is always an advantage, we don't want to discuss that today. But of course, when I compare the pipeline that in silico medicine has developed compared to, let's say, competitors in that field, what the heck was the secret sauce to get that pipeline compared to other people?
Great question. We envisioned our platform to cover all stages of drug discovery end to end. I think that's one of the core differentiating features of the platform, because we do target discovery first, deriving insights from omics data, prioritizing, ranking the targets, biological mechanisms for a disease, for a phenotype. And then we have a pipeline to generate molecules to modulate that mechanism in the right direction. So I think that's kind of holistic approach to combining biology and chemistry together helped us to accelerate. And in general, generative AI is very good at multi-parametric optimization. So what we see from our experience, the reason why we're able to speed up our programs is because we have to actually make fewer molecules. We have to make fewer iterations in the drug discovery cycle. For our program 1 in lung fibrosis, which is currently in phase 2 in US and in China, we have 2 trials running. We synthesized less than 80 molecules to get to a clinical candidate. Typically in the industry, on a good day or year, they synthesize like a couple thousand or 3,000 if you take like big pharma, very good big pharma like Lilly. So the number of molecules, it matters, and the cost and time, it actually comes from a number of iterations. If you're looking for a needle in a haystack, you will spend more time doing that, and sometimes that needle wouldn't work great in clinic, right? But if you were generating needles with right properties, right length, thickness, and quality, then you'll have a higher chance of moving faster. So generative AI helped us to be more precise, have fewer iterations in general. And if you compare to kind of the entire field, they mostly specialize in something like either chemistry or biology. I mean, they don't have a second component or third component. So that, I think, what differentiates us, because we cover it end to end. And the only 2 criteria that should be used to assess the AI-powered drug discovery company is the first, whether that company has a software, available software that others can verify, use, and apply to their problems. Not like there are like handful of companies that have software. And the second criteria is a pipeline, right? Whether they've progressed, they actually use their platform to nominate clinical candidates and progress into human clinical trials. Everything else is irrelevant. And besides the point is to create medicines. And if you don't have actual molecules in a clinic, what are you talking about? So it's the single most important criteria that should be used.
Okay, so that has a philosophical but a very practical implication, because that sounds a little bit like a Pareto distribution. Well, that's 80-20 thing. But on the other hand, when you see that high variability of human biology, of multi-omics data classes, with evolution, from the background of evolution, one would expect that the thing has that high dimensionality, that curse of dimensionality, where people are not very happy with statistical learning theory, but maths can't be cheated. So Martin, how do you see that? Is that a Pareto with the chosen few, the Pareto distribution with the chosen few, or they simply found some kind of algorithm to have a special order, but it's still a high dimensional issue?
Okay. The first challenge I see is actually that when we talk about, interfering with a pathophysiology development or a disease development, so to speak, we need actually to understand that progression over time. When you look at the data that are available, we have lots of snapshot data. We have very little, at least in the domains that I understand. I have always to say I work with neurologists and psychiatrists. So, but there you are happy if you have longitudinal follow-up data, and that's they are just a fraction of the overall data. So typically, most likely, you train anyway on snapshot data,
Yeah.
and you have no good understanding of the continuum over time, and that is besides all the high dimensionality, you're ignoring typically one of the most crucial dimensions that we have, which is the before and after, or the change in time. So I'm a bit skeptical. I see all these wonderful, excellent predictions, and that's really impressive. I see a big challenge in mobilizing the right training material. That's what we discussed already. I see a big challenge in the longitudinality and in the relevance with the real world, because most of our data have been generated in artificial settings, or I don't want to go into this mouse business, but in the neurology, neurodegeneration field, rodents again and again prove that they are not humans. So, yeah, but that was also misleading and to a large extent. And the other thing, and that's one of my concerns, is that we do not only see a flood of knowledge graphs, for instance, that are coming up now, we also see a real flood of predictive models that would probably need a sort of benchmarking or a challenge where people, in bioinformatics we have been very successful in organizing public challenges, grand challenges. I think we need something like that in the AI field as well. And in particular when it translates into medicine, into clinical practice. I just want to point out that Florian Markowitz recently wrote this nice paper with the really challenging title, "All models are wrong and yours are useless," about the lack of translation. Florian is in Cambridge, UK, and AI in oncology. And he was essentially making the point that there is a flood of models, but they die actually all on the way into clinical practice. And there is another death valley. So pharmacology, drug discovery is a bit of a different story. It's more rational because you can do an assay and can test and then you confirm. But clinical practice is a completely different dimension.
So, okay, coming to the end, I think we sang the song of rationalism during the last half an hour. Nevertheless, on the one hand, we have that multi-omics thing with hopefully promising new data classes and insights. On the other hand, we have that many kind of challenges. Do you have something, or do you see some promising breakthroughs at the horizon, so that there's not only rationalism, but also, let's say, some kind of vision, hope, or even passion, where you say, okay, there could be a breakthrough. I would expect a breakthrough in a certain area. Alex.
Well, I'm not an oracle, but I can make guesses.
You run prediction models.
And generative models too. Yeah, I would say there are a few directions that look promising to us. Obviously, I already mentioned newer generation of foundational models that can capture biology more accurately and at more fundamental level, to the point Martin made about capturing post-translational modifications and go into microRNA regulation, not coding RNAs regulation. So it's entire kind of like a dark forest, so to speak. Okay. So foundational models will evolve, will be better. About a month ago, I spoke to CEO of Moderna, Stefan Bancel, and he's convinced that within 5 years, we'll have a model with a basic understanding of biology, biological processes, so the issues we are facing now would become trivial, and the problems, right? So there are true believers from the field in the power of large language models. So that's one direction. Second, I would say we have been early adopters of laboratory automation. So robotics in this industry, it not only eliminates bias, human bias, human error, but it also brings a lot of like quality and performance-wise, it's far more performant. You know, robots don't take vacations. They're not like they work 24/7, right? So automation, laboratory automation, will bring not only better quality data, but also will bring more data out. And we have a lab which is fully automated, 800 square meters of lab space. So I think we'll, like, this automation is inevitable, and all industries will be forced to automate or will become irrelevant. And third, promising direction that we are tapping into is quantum computing. Last year we published 2 papers on quantum computing, and this year we published the first paper where we use quantum computing, like real IBM quantum machine, to generate real molecule against KRAS protein. Well, it's famous target, previously considered like undruggable or one of the harder targets. But we generated a molecule using quantum machine, and it turned out to be a real binder to the KRAS with a completely new chemotype, right? I think within 5 years or around that, we'll see more applications of quantum computing, and with the advent of more and more prominent machines from IBM, IonQ, and other providers, we'll see probably a breakthrough in that space too.
Okay, thank you, Martin. Beyond rationalism, where are promising breakthroughs? Where can we expect something?
So, as I said, I would actually see the realistic option that AI is supporting the discovery process or the consolidating process when it comes to knowledge and data, assessing data in a rational way, the data quality and so on, supporting the classification of knowledge into independently validated or rather fuzzy or uncertain. So certainty, uncertainty in data and in knowledge, and helping us to move discovery actually forward in a more and more AI-supported science process. I think that is where I see the breakthrough actually, that our classical understanding of how we do science is undergoing a massive change, and I think we should embrace that and welcome it. That's it.
Thank you very much. Thanks for listening us. That's it for today, and watch out for our next program point. Thank you. Bye.